crops through remote sensing data. So my topic outline will be the outline slide is okay, right? You can see slide movement.
Yeah, yeah, I can. We can see. Yeah.
Perfect, perfect. The slide movement is okay. So my talk outline will be, I will be talking a little bit about my background, my research background, and when I came, where I came from, and where I am, and then I will talk about little bit of basics of remote sensing and how the remote sensing principles and tools and techniques is used for agronomic management, and then I will talk on future challenges, and at the end we will have a couple of questions, and we will talk more formally or informally. So my background, where I'm from. So I'm basically from the state of Bihar, where I started my schooling, and then I moved to Banaras Hindu University for my agriculture sciences agronomy undergrad. After undergrad, I moved to IRI, Environmental Sciences Department, where my major was environmental sciences, minor was agriculture, physics, and climatology. And I did my master's from IRI in New Delhi. And then I moved to US at Michigan State University for crop and soil sciences PhD program, and the focus was crop and climate modeling. Before masters and PhD, I worked few months with Indian Institute of Soybean Research as a geospatial crop modeler, and then I started my professional journey at Kansas State at postdoctoral research fellow. And my role was a geospatial crop modeler there, where I worked on remote sensing, crop modeling, and recently I have been promoted to associate scientist at Feed the Future Universal Lab, where we work mostly for African countries, which I will talk more on a couple of slides as well. And very recently, I accepted an offer for assistant professor at Mississippi State University, where I will be joining this summer. And this position is about agriculture climatology. This is my journey where I came from and then where I am right now. Before I go to a little bit of principle of remote sensing, and then I will come to use of remote sensing or application of remote sensing in agriculture, let's have a small video so that you will have a glimpse of what kind of technology tools as well as principles we use for agriculture or use in agriculture. So little bit of video for 30 to 40 seconds, and then we'll come on basic principles. So if you see here, the satellite imaging can capture all those development at the land, which can be vegetative development or any land use changes or any kind of system in the land where we observe. For agriculture, especially what we use those data to georeference to collect the farm, and this is a case study for a farm in Punjab. You can collect a lot of indices, and based on those indices you can do a lot of analysis based on land use history, based on historical map or yield map. We can do multiple things on organic farming, regenerative farming, crop identification, depending on what kind of use we are aspiring for. If you are an engineer, your use will be different. If you're an agronomist, your use will be different. If you're a hydrologist, your use will be different. If you're a climate change expert, you will analyze greenhouse gas emissions on different layers, so that you can do those kind of parameters. So based on stakeholders use, remote sensing can be used in agriculture for multiple purpose, starting from dashboard to analyze that kind. So this is a brief analysis what we can do remote sensing. So right now, if you just saw a video where you saw what kind of analysis we can do at farm, from farm scale to landscape scale, and we can do multiple analysis. But how we can do that? So just for an example, do you know that our eyes are also remote sensing organ? When we see something from remotely and we analyze in our mind, that is processor there, right? So that's why we call our eye as a remote sensing organ. And then we analyze everything, and then we perceive that what is the object, what is the differences, and that's how the preliminary remote sensing organ in our body is eyes. When we talk about physical science, where we work on remote sensing bodies or remote sensing data assimilation, what we talk about? We talk about anything, object or item or anything at a distance where we can sense it using some kind of photographic image, some kind of radiation, some kind of wavelength, some kind of any kind of sensing medium. We can say that we can get that thing at the remote place. That is called remote sensing, and different kind of sensors are used for different kind of applications. The goal of stakeholders are, what information do you need? based on your requirement, your sensors, your satellite data, your tools and techniques will be different. And then again, how much detail. For an agronomist, detail, or engineer, agriculture engineer, detail is very important. Once we work on how much resolution of space or time, then we think of how much detail we need. And then we talk about how frequently you need the data. When that is called temporal resolution, we will talk that as well. So these are three basic things for agronomist or agriculture scientist, where we can think of what, how much, and how frequently, so that we can plan. And that's how we call agronomic management. So for agriculture, we talk always about G into E into M, genotype into environment into management. So genotype is a characteristic of varieties, environment, our environmental variables, weather variables, and then management is the factor. So E into M interaction is very important, and that's how remote sensing data helps lot into E into M interaction. So how we do? So naturally, remote sensing, I talked, we can see, sense, perceive, and we analyze on the brain. But this is called natural remote sensing. But how far our eye can see? Our eye can see a certain distance or certain resolution where we can analyze the data. But when we try to look for distant object, we need to in normal language we say zoom in or zoom out to get that point. For that point, how we can get that? That is called artificial remote sensing, and that's how a remote object can be sensed through sensors on the satellite or on any tool or US, and that's how we can analyze that information in a software, in a tools, and extract those information to make a meaningful action. So one thing is very important here: data and information are useful, but for agronomists we make it usable. So useful to usability is a very is a thin line difference between useful and usability. So data analyzed through remote sensing are useful, but for agronomists, we should make it usable so that we can get real information at the ground. And that's how artificial remote sensing helps in converting useful data into usable data. And that's how we always confuse between what is proximal, what is remote sensing. So there's a basic difference between remote sensing and proximal sensing. Remote sensing is something which is have an object from a distance. Proximal is little bit closer. When we say proximal means either you can get into contact or a few meters away. At the field condition, for agronomists, we have a lot of tools and techniques which is proximal sensing, and remote sensing data we usually acquire and we analyze for those data sets. So what are the difference between proximal sensing and remote sensing? At the field, we have a lot of sensors which can be considered as a proximal sensing. So you can see these kind of images here. And in the crop physiology or stress experiment, we do a lot of sensing of eddy covariance at the field or LICO data at the field. These are called proximal sensing, where we can have a physical contact or few meters away. But the remote sensing, basically, when we work on large area field to landscape analysis, then we need to work on remote sensing. And that's how, if you see, they are the ground-based sensing, remote sensing tools, and some are the aerial remote sensing tools. The satellite, fixed wing, UAS, unmanned aerial system, that is called drone popularly, those are called aerial remote sensing tools, depending on how we use that. So basically, in the remote sensing section, we usually work on what kind of wavelength we use. So in a couple of slides, in few more slides, I will talk more about those spectral and reflectance concept, where we need to work on what kind of resources we have and what is our target study area or target study uh, mm, area means zone. And that's how remote sensing is classified in different areas. So based on wavelength, the optical, thermal, infrared, microwave, and then optical also classified into panchromatic, multispectral, hyperspectral, and then in the microwave, active and passive. So we'll talk more about that in the later slides, but this is the brief of what kind of data we use. And this is the what, and then how frequently, or how fine, or how precisely we use that. When we say how frequently, that is called temporal resolution. When we say temporal resolution, how what is the duration of visit of that data sets. If you want everything on 5 day interval, or if you want everything in 1 day interval, so the time gap is usually called as a temporal resolution. And I won't I will not go one by one because I will share that slide you will have. The point here is that the temporal resolution means the changes. Let's say if you're working on irrigation parameters, you need to work on temporal parameter of at least 4 to 5 days so that you can have water requirement at critical growth stages. If you are analyzing land use change, or if you're analyzing biomass change, the pattern will change. So based on what kind of criteria you are working on, the temporal resolution will change. Similarly, we have a spectral resolution. So what kind of sensors we are choosing, with what interval, wavelength interval, and then that's how we choose different kind of sensors or satellite database. So if you have heard about Landsat, MODIS, and other data sets where we choose different spectral resolution as well. And then there is a spatial resolution, what how much bandwidth you need to, we have 500 to 500 meter, or 30 meter to 30 meter, or Sentinel, or if you go to Pleiada from French, or Planet, those they have 1 meter resolution as well. So based on user requirement. And the third one is a radiometric resolution. And basically for radiometric resolution, we need to have differences between pixel to pixel, and then the sensor's ability to discriminate differences in that pixel. And that's how we have different kind of sensors and different kind of radiometric resolution on the sensors as well. And these are the three basic kind of resolutions for any kind of satellite data we work on the remote sensing. Now, what is the basic? So I will come back on the basic of um, remote sensing work, or any kind of UAV or satellite data work in agriculture field. So basically, when we see anything, the visible light range is 400 to 700 nanometer, right? And if you see for different objects, they have different wavelength, they have different energy, and they have different temperature related to that energy scale. And that's how, as a stakeholder, as a user, we need to identify what area we are working on, right? So basically, when we work on agriculture field, we work on mostly reflectance data from 350 nanometer to 2450, right? Here, what you see here, you see here the reflectance. So y-axis is reflectance in terms of percentage, and the wavelength is x-axis here. So this is the presentation representation of what we see at the field. When I say what we see, means instrument or sensor, right? They usually capture those kind of data sets based on the reflectivity or reflectance, we say. That is the fraction of incident radiation versus reflected by that surface. And if you see a lot of peaks and valleys here, right? Those peaks and valleys depending on how much they observe, and then how much absorb, and how much they reflect there. So basically, for any remote sensing expert, they work on how they analyze those data set into different field. So basically, for vegetation, because we are in agriculture sciences, we work on agronomy, agronomical management. We are talking today, so we work on vegetation, soil, and water. These are three major components where we work on, and that's how you see these are the different measurable data, which is a function of wavelength, and it's called spectral reflectance in certain wavelength. That's how we usually measure that. For leaf, basically, if you see the peak area of the green band, which is reflected by chlorophyll in the visible area, 400 to 700 nanometer, and then the blue and red band. So basically, what you see here the reflectance by the leaf structure. So for the vegetation especially, the leaf structure has different component. They have chloroplast, they have mesophyll cells, and especially in the case of vegetation, the light absorption by leaf pigments, especially they dominate the reflectance spectrum in the visible range. When I say visible range, that is called 400 to 700 nanometer. So the chlorophyll pigments, if you know about the pigments A and B, the chlorophyll pigments A and B selectively absorbs blue. When I say blue, that is 400 to 500 nanometer. And then at the red side, that is called 600 to 700 nanometer. So these are the wavelength good for photosynthesis. We know that in crop physiology courses or in basic agronomy courses, we read about that. So the chlorophyll pigments A and B selectively, absorbs blue and red lights for photosynthesis, and there is a less absorption over the green wavelength. If you see that the peak one, 500 to 600 nanometer, and that peak is more reflectance based on, that's why we see leaves are green, right? And that's the green appearance of all those healthy vegetations. For agronomist, we basically focus on how much there is a green or how much there is a healthiness of the leaf, right? And the pigment, if you see the yellow or orange red pigment, which is called carotene, has a strong absorption in the blue wavelength, 400 to 500 nanometer. So the red and the blue pigment, xanthophyll, uh, they also absorb strongly in the nano range of 400 to 500 range. So my point here is that reflectance of green band in the visual range is a signal where we highlight that leaf is more green, it means more healthy. If it is not healthy, it means it's stressed, and it could be a stress for disease, nutrition, water, anything. So stress vegetation will give off a different spectral signature. The spectral signature is a word we use for the spectral pattern, right? So the stress vegetation will give different kind of a spectral signature corresponding to the effect of stress. It could be disease, it could be pigment degradation, it could be any kind of stress condition, right? So that's how most of the energy, if you see, is transmitted and reflected depending on leaf structure, mesophyll to leaf chloroplast area, and then that's how different kind of infectance you see. In the figure I explain about, in the figure I mentioned here, see what kind of cuticle, upper epidermis, and palisade cell and mesophyll cells, how they reflect. So based on those reflection, we analyze those reflectance percentage on different layers of the leaf, and that's how it reflects